our cells have a need to transport nutrients and proteins from one location to the next location. How are these protein molecules transported from where it is synthesized to the location where it is required? That is what we are going to discuss now. We have seen in the earlier video that this intracellular trafficking is coordinated with the help of vesicles. And there are three kinds of vesicles. That is what we have seen. COP1, COP2 and clathrin coated vesicles. These are the three types of vesicle. In this particular video, we are going to focus on especially in the COP2 vesicles. Now, all these vesicles requires a kind of a G protein binding protein. COP2 has a G protein binding protein that is called SAR1. So SAR1 is a G protein binding regulatory protein. Now this SAR1, it can bind to GDP. If it is binding to GDP, it will remain in the cytosol. If it is binding to GTP molecule, GTP bound SAR1, this is membrane bound. So this is one point that we need to remember. Now, what is in the first step in transporting the cargo? So that is the formation of a vesicle. Now, formation of a vesicle is initiated by this SAR protein. So let us diagrammatically represent this entire process. Let us assume this is the SAR protein. Okay, this is the SAR protein and this is bound to GDP molecule. GDP bound SAR, so therefore it has to be in the cytosol. If the initiation comes when a cargo has to be transported, what will happen is there is a membrane bound GTPase enzyme that is known as Sec12. Now, the Sec12 is nothing but it is an SAR that is GEF. It is SAR GEF. GEF stands for guanosine nucleotide exchange factor. That is a GTPase. So what this will do is it will replace the GTP, it will replace the GDP with GDP is removed, so it will replace this GDP with a GTP. Let us make the correction here itself. So this is now being replaced with a GTP molecule. Now this is carried out in the GEF protein. Once it is binding to a GTP molecule, now there is a change in the charge. So because GTP it has got a more negative charge in comparison to GDP molecules. Because of the negative charge, there is a slight change happening in this SAR protein. What is the change happening? It will expose an N-terminus. It will expose an N-terminus region in this protein. That N-terminus which is hydrophobic, and this hydrophobic region will get inserted into the membrane of the endoplasmic reticulum. Assume this is the endoplasmic reticulum membrane, this is the cytosolic side, this is the lumen side. Now this GTP bound, this is inserting itself into the membrane. This portion is the hydrophobic portion that is inserting itself. Now this is the first step that is happening in initiation. Once this initiation occurs, now it has to be converted into a bud, it has to be converted into a vesicle and pinches off. Before it gets converted into a vesicle, cargo must be selected. Let us discuss about selection of cargo. So we will leave this topic here and we will come back to this topic. What happens, and after looking at how the cargo is selected. Let us look at how cargo is selected to be transported through vesicles. So understand that the cargoes are mainly, it is the protein, and normally only a fully assembled protein or fully folded protein is selected. If it is a protein with the multi subunits, all the subunits must come together, it has to be assembled, then only a protein is transported. So improperly folded proteins etc. are not transported through this mechanism. Now a protein can be transported through two mechanisms. In the two mechanism, one is called a bulk flow, and the second mechanism is known as the signal sorting. So these are the two mechanisms used for cargo selection. Bulk flow, assume if this is the protein, okay, these are the proteins, and these proteins are directly inserted into either forming vesicle. So in the vesicle, these are directly taken inside. That is bulk flow. In the case of signal sorting, if this is a protein that has to be transported, now these proteins will have a signal present. So if the signal is present here, now the signal will say where it has to be directed. Based on the signal, if the proteins are selected into the cargo. So therefore, you can see that here is a protein that is selected based on the signal. It is based on the signal it is selected. So these are the two ways by which cargo is selected. Let us look at the signals also. So elaborate research has resulted into understanding in the nature of these signals. The kind of signals that are found is, one of them is diacidic signals. The first one is diacidic signal. So what are these diacidic signals? For example, these proteins will have a transmembrane region, then many amino acids, and the sequences etc. have been verified. Then it will find the acidic amino acids like aspartic acid, then there is any one amino acid, then there is an aspartic acid, then it is continuing with a few more amino acid, then ends in the C terminus. So this particular combination is called the diacidic signal which is encountered, and this signal is mainly seen in COP1 mediated transport. Another kind of signal that is commonly found is diphenylalanine signal. So in this kind of signal, so in the transmembrane region, then about 10 very specific amino acid followed by a phenylalanine, 2 phenylalanines that ends in the C terminus of a protein to be transported. A third kind of signal which is found especially in proteins that has to be embedded in the transmembrane region, and that is a type 1. Type 1, you know that the N terminus of the protein will be in the lumen. So such proteins will have a kind of signal. So transmembrane region, number of amino acids will be there, followed by lysine residues. So you have a lysine residues, then an X, a lysine residues, then there are 2 threonine residues followed by 2 threonine residues, 2 and followed by the C terminus. Another kind of a signal. Another kind of a signal, a fourth one, that's the last one we shall see. Another kind of signal that is meant for the type 2 transmembrane proteins. Type 2, you know that the C terminus of the protein will be in the lumen. That will have signals like number of amino acids followed by arginine. It's called diarginine. That will be at the N terminus of the protein. What is important for us to see is how different arrangement of amino acid giving a very specific signal, and this signal is used for transporting a protein from one location to another location. Many of these sequences have been identified and they have been characterized in many species. That is the source of our information in order to understand our knowledge of intracellular trafficking today. Let us proceed to understand how the vesicle formation occurs. We have seen in the first step, if this is the endoplasmic reticulum membrane, so our GTP bound SAR1 is embedded in the membrane. So it is embedded in the membrane. We have also looked at how the cargo selection procedure occurs. In order to achieve that, now what has to be done is it has to form a kind of a curvature in the membrane. It has to bend. Now this bending is mediated by this SAR protein. This is SAR1 GTP bound SAR protein. It initiates the process. Now once it initiates the process like this, it will bend. Once it initiates the process, now what is going to happen is it has to be coated with the coat proteins. Now if the coat proteins is very, in order to bring them into this, we need to look at an interesting aspect about the structure of membrane. Every membrane, roughly about less than 10% of this membrane has got phosphatidylinositol. We know that phosphatidylinositol is a phospholipid. Now phosphatidylinositol can be phosphorylated at 3 different places, and these 3 places are 3, 4, 5. That is important for us today in order to understand this. Now this phosphatidylinositol, if it is phosphorylated at the 3rd place, we will say that phosphatidylinositol at the 3rd place it is phosphorylated. If it is phosphorylated at the 5th place, we say phosphatidylinositol 5th place phosphorylated. If it is phosphorylated at the 4th place, okay, phosphatidylinositol at the 4th place it is phosphorylated. So therefore there are definite phosphatidylinositol kinases present. They will phosphorylate this. Now once you have 3, it can be phosphorylated into phosphatidylinositol 3,5-phosphate. This can be phosphorylated into phosphatidyl also 3,5-phosphate by another kinase. Now these can be phosphorylated into phosphatidyl 3, 4, 5 phosphates. This way it can be phosphorylated. This can be phosphorylated into phosphatidyl 3,5-phosphate in this way. Same number of phosphatases are also present in a cell, because depending on the changing need of the cell, these phosphatidylinositol they will be synthesized depending on the need of the cell. We know that if you remove a phosphatase, if you remove the phosphate, it will become phosphatidylinositol 5-phosphate. So similarly, this can be done this way, this can be done this way. You can remove the phosphate depending on the need, whichever phosphatidylinositol is needed, that is being synthesized. This is an interesting aspect that we need to keep in mind as we proceed with understanding the formation of vesicles. Now this plays a crucial role. Phosphatidylinositol plays a crucial role in bringing the coat proteins to the surrounding. So in the coat proteins, there are many coat proteins, especially when we are talking about COP2 coat proteins. Major component is a coat protein called SEC23 and SEC24. These are the 2 important coat proteins. These 2 coat proteins forms the inner coat. It forms the inner coat. Let us see how the coat is being arranged. Now this is the cytosolic side, this is the lumen side. If the protein is here and normally the proteins that has to be assembled into the vesicle will be normally getting closer to the exit point of the endoplasmic reticulum. This is the endoplasmic reticulum, and they will get assembled here. A curvature occurs now. So a curvature occurs in this manner. Curvature occurs in this manner because of this GTP bound SAR1. Now the cord proteins come into picture. So assume this is a sec protein. Okay, this is a sec protein, sec 23 and 24, sec 23, 24. This gets added to this GTP bound SAR1. So we can represent that in this manner. So here is a SAR, GTP bound SAR, GTP bound, and to this, the cord proteins slowly starts getting assembled, one after the other. And through all the possible interactions, it forms a coat around this. This is what we call the inner coat. The inner coat is formed with the help of sec proteins 23 and 24. Along with this inner coat, there is an outer coat is also formed. Now the outer coat is formed with the help of two other sec proteins, and they are sec 13 and sec 31. We will come back to sec 31 later on. So sec 13 and 31 forms an outer coat, and that outer coat is formed around this. So you have an inner coat which is red in color, and you have an outer coat which is green in color. It has already taken the cargo inside. We have looked at the procedure by which cargo is taken inside. Now what is important is the next step. That is step number 3. Step number 3 is to pinch off this. That is known as step number 3 is fission. Okay, step number 3 is fission. Step number 1 is the cargo selection. Step number 2 is the formation of the vesicle, and step number 3 is the fission. How exactly this gets removed in the case of cord protein 2 is not yet very clear. So this gets removed, and now the vesicle is free. Now the vesicle it's free, and it is in the cytoplasm. It can be transported to a location. Now, in the next step is, now this vesicle must, that is step number 4, the vesicle must get fused with the membrane. So this is the vesicle that is formed, that got pinched off. Two important proteins get associated when the vesicle is formed. These two proteins are, one is called a RAB GTP. It has an important role. RAB GTP is one of the proteins that will get associated with this vesicle. Another protein is called a snare protein. We will be looking at the snare protein elaborately a little later. The snare proteins, since it is associated with the vesicle, we call it V-snare protein. So now the protein in the vesicle is ready. Now we come to step number 4. So step number 4 is uncoating. Why uncoating is needed? So this will remove. Why uncoating is needed? Uncoating is needed then only this membrane is capable of binding to an acceptor membrane and getting fused. So this outer coat, inner coat must be removed. It is very easy to remove the outer and the inner coat. We have a protein in the cytosol that is called the SAR-GAP protein. GAP stands for guanosine nucleotide activating protein. That is nothing but a GTPase. So this will remove the GTP which is bound to SAR. So a GDP is added, a GDP is added, GTP is removed. Now you will have an SAR bound, GDP bound SAR protein. You know that it has no affinity towards in the membrane, it will get separated, it is supposed to be in the cytoplasm. Along with that, the coat proteins also get removed. The inner coat proteins as well as the outer coat proteins get removed. What is left now? It is only this in the naked vesicle. In the naked vesicle with these two proteins which we have mentioned, those two proteins, one is the RAB GTP proteins, another one is the snare proteins. Yes, NARE, snare proteins. So this is the protein which is supposed to be merged with the acceptor membrane. So that is the next step. That is step number 5. Step number 5 is once the uncoating occurs, step number 5 is a process called tethering. In fact, step number 5 and 6 join together. Step number 6 is called docking. This is step number 6, tethering and docking. How is tethering carried out? Tethering means it is to connect, it is to link, establish the first connection. So here we have the protein, the vesicle with the cargo, and this has the RAB GTP. This is RAB GTP. And you also have the snare protein. The first step in tethering is on the membrane here. This is the acceptor membrane. This is the acceptor membrane. This acceptor membrane has a protein called, it is called the tethering protein. And the tethering protein is a RAB receptor. So it has a RAB receptor. Now this RAB receptor will bind to this RAB GTP. It will bind to this RAB GTP and it will bring the vesicle close to it. It will bring the vesicle close to it, establish the first primary connection is established. Once the primary connection is established, remember it has got the snare protein, the V snare protein. Now, in this acceptor membrane also has an equivalent snare protein that is called the T snare. The T snare has got 3 chains. Okay, the T snare. So let us draw the T snare. Assume this is the T snare with the 3 chains. What is going to happen now is this vesicle, in order to merge, it must be brought about 1.5 nanometer close by. Now, in order to bring it close to about 1.5 nanometer, it is a hydrophilic environment. Water molecules must be removed from here. And removal of water molecule is thermodynamically not feasible. And this is facilitated by the binding of the RAB receptor or effector, RAB effector. So this binding that gives the energy in order to remove water, and the binding can become stable and stronger. So what is going to happen is this is brought close by. So let us draw this. Here is the V snare, and that will combine with the V snare. So it forms a kind of a helical structure. It forms a kind of a helical structure, and it forms a 4 helical wrap. It forms a 4 helical wrap is formed, and here is our vesicle, and here is also one more, the V snare, and here is a T snare merging with that, 1, 2 and 3, it is formed, and it has already linking with the RAB proteins, and these are the RAB effector. So this is the next diagram. So let us remove all these. So therefore this has the cargo, a linkage is established, they are brought together. This bringing together, it is associated, they assume that this pulling is with the help of this RAB effector protein is like a fishing line. it hooks in the vesicle and pull it close to it, maybe by using actin or microtubule based transportation. It is brought inside with this kind of a motor and now it is ready to be fused. That leads to the next step. That the next step is step number 7, that is the fusion. So in the fusion, in order to have the fusion, what needs to be done is this Rab proteins must be removed. Then only fusion can occur. Now, in order to remove the Rab protein, so Rab, it is a GTP bound. Remove the GTP, so Rab GDP bound Rab is removed. So the whole thing gets dissociated. Okay, and this will get, therefore it will promote the merging. Now, if the Rab GDP is binding to, a protein known as the Rab dissociation inhibitor. Rab dissociation inhibitor, it binds. So this is the red one is the Rab dissociation inhibitor, which makes the Rab protein soluble and in an inactive state in the cytoplasm, which is very important. So once it does that, now this will get merged. It will get merged if the cellular contents are released into the location where it has to be released. So for example, if it is a Golgi bodies, it is released into the lumen of the Golgi bodies, and this membrane partly it gets merged. So that merging, let us see how this occurs. So assume this is the membrane of the vesicle and this is the membrane of the Golgi bodies. This has got 2 layers. What is going to happen is, initially this forms the membrane of the vesicle and these 2 membranes join together. This is the membrane of the Golgi bodies, this is of the vesicle. These 2 join together. Then what happens is, it forms like this. And the contents are passed into the lumen of the Golgi apparatus. So these 3 diagrams explain how exactly the fusion occurs. So once the fusion occurs, what is important is, if you look at the membrane, here you have the tethering proteins and the tethering the snare proteins. If the snare proteins have been linked with the V snare, it has to be separated and it has to be brought back. Now this dissociation will ensure that this is getting separated, and once it gets separated, so the remnants will be transported back through the retrograde transport which we have learned, and through the retrograde transport by using COP1 protein vesicle, it will be brought back into the endoplasmic reticulum or back into the lysosomes. Before we look at the last step of this transportation process, let us look at the snare. What exactly is the snare protein? So when scientists were studying this mechanism, the 2 important scientists who carried out a lot of work in understanding intracellular vesicle mediated transport is, one is G. Pallard, somewhere in the 1960s, and later on by Randy Schekman. Both of them won Nobel Prize for the kind of work they have carried out. So let us look at what exactly is the snare proteins. Now, how this name comes snare? They were looking at a particular chemical, and the chemical name is N-ethyl malamide. So N-ethyl malamide, so therefore the short form is NEM, N-ethyl malamide. So it binds to sulfhydryl groups and can inhibit a protein. So this chemical they were using in order to study certain mechanisms. Now, a protein which is sensitive, that is NEM sensitive factor, that is called NSF, NEM sensitive factor. What is the snare? Snare is nothing but soluble NSF attachment protein receptor. So that is what is snare. So soluble NSF attachment receptor is snare. Now we are going to come across another protein that is called SNAP protein. That is soluble NSF attachment protein. And we will see what is its role in the last step of this intracellular trafficking by using COP2. And the last step, that is the 8th step, is the recycling. So in order to recycle, especially in the V snare, we have a diagram right now. So that is, the vesicle has been fused and the contents have been released, and we have the T snare, which is forming a kind of a link with the V snare. This has to be separated. How to separate this? It is in order to separate that we have the SNAP protein. We have just seen the SNAP protein. The SNAP protein along with the NSF. SNAP protein, NSF is a hexamer. NSF along with the SNAP protein that will act as a GTPase. So this will remove in the bound RAB proteins. Now the RAB is removed with the GDP, it is removed, and it also removes the snare proteins. The snare proteins are removed. Now this separation will ensure that now these proteins, RAB GDP, as soon as RAB GDP is released, RAB GDP will combine with the RAB GDP. It will combine with the RAB GDP inhibitor. Okay, so the RAB dissociation inhibitor will bind with this. RAB dissociation inhibitor binds to this, and that will ensure that the RAB GDP is soluble and as well as it is inactive in the cytoplasm. So now the snare proteins that can be picked up along with the membranes by the COP1 coated vesicles, and they are transported back into the endoplasmic reticulum in order to carry out the next round of reactions. So this is how the entire process of intracellular transport happening with the help of COP2 coated vesicles.